Good morning, my name's Olivia Fulton and I'm one of the patient chairs within SHARP, and I'm here with Jacques Bouy from Teva, and we're interviewing Ian Adcock today, a biochemist from Imperial College in London, who's also one of the leads in Biopret. So good morning, Ian.
Good morning, Olivia.
I just want to start with, what was it that made you want to get involved with the SHARP project?
After spending time with Biopret, where we looked at a small number of severe asthmatic patients across Europe and went into huge depth trying to understand a lot of the complex biology that goes in, it became very clear that some of the things we were looking at really weren't connecting to what we saw in relation to patient outcomes. So, for example, the molecular biology, the gene transcriptomics, was not picking up PROs in our cohort. And was this going to impact across the whole of the severe asthma across Europe? So for me, SHARP became a logical extension of that deep phenotyping to look at the whole of Europe. Is severe asthma the same across the whole of Europe? Are there differences depending upon the healthcare system that's involved or in the way that the patients are diagnosed? So I think that the questions and some of the failure to address our questions made it essential that we looked further as part of Biopret. SHARP came up as really the ideal platform for us to try and work together with patients, because we had also realised through Biopret that patient engagement was critically important. It was something that was new to me 10, 15 years ago when we started, that patients actually play a critically important role in the development of projects and addressing questions. So to my mind, lots of tick boxes were filled by SHARP. And so it was a really great project to be involved with.
Thank you very much, Ian. Can you give a little bit more insight what Biopret exactly is?
Yes, Biopret stands for the unbiased biomarkers for the prediction of respiratory disease outcomes. It was initially going to look at both severe asthma and COPD, but of course money runs out. So it was focused quite correctly, I think, at the time on severe asthma. So this was a pan-European consortia of about 20 academic centres, about 9 or 10 companies. It was a big project to try and understand severe asthma.
And if you look at the long term, where do you really see these synergies?
I see the first synergy, and I think is probably the most important thing, is the recognition of how we as basic scientists and clinicians and patients need to work together, because this is not myself working in a lab playing with cells. It's not a clinician seeing a patient and the patient coming in to clinic. This is all of us working together, and it's only working together will we be able to make step changes in our understanding of disease and treatment of patients, which is the ultimate goal. I do not feel that we should remain in the stage where patients are not treated as well as they should be, because we either on a cell-based assay haven't seen something, or on a you come into a clinic and things aren't as perfect as you'd like to be. I think we just really need to work as a team, and I think this is how SHARP is now at the pinnacle as far as I can see. But it started for me with the work in Biopret, and hopefully this will drive through for the rest of my research career, is just how important this synergistic collaboration is, and how it has changed and will change in future our way of looking at disease.
Has it changed at all the way you work alongside patients?
Personally, yes, it has, because I think it made me recognise that we do have an extra expert in the room when we talk about this, and that's the patient. What is a patient? When I look at mean data. The mean patient or average patient just doesn't exist. And this is where I think Sharp has really emphasized that patients are individuals. They need to be treated as individuals. We used to make these allusions to personalized medicine. Yet the one person that it was really important for, we didn't talk to. And that just seemed crazy. And now, having been engaged with Sharp and initially with you, Biopret, you recognize that the most important person, if we're going to do personalized medicine, is the patient themselves. And talking to a patient, you find out so much more about their disease rather than asking them to fill in a huge number of checklists when they come into the clinic, give numbers of samples. That's not the patient. And to me, it's really exciting how we see things. I did mention something about PROs, the patient-reported outcomes. I suggested that one of the things we could do is to look at PROs that we have in New Biopred and say, does that link with any of the omics data sets that we have? In some ways, I was disappointed that we didn't see something obvious with transcriptomics, but we're seeing something on metabolomics and eicosanoids, which is the downstream effect of the whole body. So these PROs are detecting something that we don't see in any other system. So I think, by working as a team and particularly working with patients, because they're also giving us further information on this, and we will come to Sharp with a proposal that we push this forward a lot more, because I personally believe that this is that merger between patients, the patient registries, and deep phenotyping. We can produce something that is really helpful to the individual patient.
Really exciting.
We've made great strides, and there's no denying that we have improved the lives of many, many patients. But there's still a huge gap. We haven't done everything we hope to achieve. And, for individual patients, they still suffer incredibly badly. It's only discussions with patients that you realize just how bad severe asthma is. My wife and some of my friends have asthma, but this isn't severe asthma. This is not life-changing asthma. And to me, that again was a revelation, just how much someone's life becomes totally controlled by a disease that we should be able to treat. So I think that we need to redefine T2 asthma. We need to redefine what isn't T2 asthma. And we need to do this in relation to patients, what makes it understandable to a patient, which means really linking it to a drug or a combination of drugs that's going to treat their disease.
There's a huge unmet need. I speak to a lot of patients in the advocacy work that I do, and it's a frustration that they get of, well, you don't qualify for these biologic treatments, but we don't have anything for you. So I think it's a really exciting part of asthma to try and be able to give this group of patients hope and something that can help them.
Yeah, I think you're right, Olivia. It's something we should do as a community involved in research, is to stop calling it T2 low, and let's be positive. And this is why patients and Sharp can make a big difference in making the community recognize that an absence of something doesn't mean you don't have a disease. It just means that we haven't labeled it, and maybe a label is the most important thing that we can do currently, because that shouldn't be so difficult.
Let's assume that Sharp had all the resources they wanted to have. When do you think then we could make a big step forward to offer almost all severe asthma patients a treatment plan so that they can improve their lives?
Yeah. That's putting me on the spot, but a wonderful question. And I think I will approach it from two sides. The first thing we really need is, for the patient, we need to have something that's easy to give as a biomarker. So a blood sample, an exhaled breath sample. We can't ask a severe asthmatic to give us a bronchial biopsy on a regular basis. This is ridiculous. So we need to have a big screen on blood and breath, but we need to then relate it with a much smaller focused group of patients where we do go into the airways. And the wonders of, I guess, molecular science now is the single cell, single nuclear spatial sequencing, where we really get to understand how cells talk to each other within the tissue. But it goes beyond that. We are running drug studies at the moment where I feel that as a community, it should be incumbent upon us to look at responder, non-responder populations, because I have a great idea. I think anti-IL-22 should be wonderful for a subgroup of patients, and it works for some patients, but not all. Do we just stop and say, it didn't work as a study trial, it's failed? Or should we say, let's understand why it's failed? And the reason we're going to understand is by looking in the site of disease, which is the airways, and use that data to say, in the blood or in my exhale breath, I can now relate that responder population to a particular biomarker and the non-responder population to another set of biomarkers. So every clinical trial, I feel we should be doing this. And that will give us much greater insight. It will give us, well, you have a set of markers that says you will respond to drug XYZ, but you're certainly not going to respond to ABC, and you might even get worse with MNLOP. So this is the way I see the future. My gut feeling is that it will be some sort of biosensor that doesn't even include taking blood samples, whether it's an eNose type device or just something we get from the skin. This is the thing I feel. It's quite sad, because for those of us of a certain age, we remember Bones in Star Trek and his tricorder that just used to determine every illness that was ever going on across every species in the whole universe. And that was essentially just smelling out organic volatiles. So I sort of still think that's going to be the way forward. But I think it's the linking of the easily accessible biomarkers with some very deep molecular phenotyping that we can now do, which wasn't available 5 years ago. But also every drug study we do, I feel we should be, as a community, saying we need to understand more. If we do a large study, it fails because we don't hit the endpoint. Is that the right endpoint we should be looking at?
So your advice is also to have an in-depth look into the non-responders studies.
Yeah, the non-responder tells us as much as the responder. And it's particularly important. I would like to know, I'm not going to be given a drug that may have potential side effects if it's not going to do any good at all to me. And it's a waste of time. Let's go on to something that might do me some good. I don't want 3 months or 6 months or a year to say whether it's done anything, because my disease is, or Olivia's disease particularly, is a year of ill health, taking a drug that's never going to work.
But that means for the pharmaceutical industry also a paradigm shift, because
it is, yes, yeah.
We usually look at the responders and we hope that the number of responders is almost 100%, which is unrealistic.
Yeah.
But I think that's very good advice that the pharmaceutical industry is also involved in SHARP, that we also will take another look at data than we're used to. So we all have to change our positions. Yeah.
No, I think that's right. I know that there are clinical studies where people have done biopsies and they're sitting in a freezer. Overall, the drug study was negative. But within that negative study, you have people who were super responders, and let's go back and have a look. It's a bit of money, but we do have data and samples. And I think if we're going to really drive our goal for the truly personalized medicine, this is what we need to do. Because otherwise, it's a waste of time and energy and cost for the drug company. But even more importantly, we're doing the patient a disservice. We need to make that mind shift.
Olivia, do you have any other questions?
It was more a sort of thought that's really struck me, just how much data about patients is actually sitting dormant in a freezer because it didn't meet the end goal of whatever study it was in. And you just sort of think, right, wonder how much it could help people, not just in asthma, but across the whole.
Every chronic disease, yeah.
Every day we get more data in the SHARP severe asthma registry. So we are collecting data, we have data in the freezer or whatever.
Yeah.
That's not the issue. The issue is mind shift of scientists, doctors, and so on, people who work at the pharmaceutical industry. I would like to thank you, Ian, for this really inspiring conversation.
I just like to say that this has been really good fun. And it's actually, whilst we've been talking, it helped clarify my thoughts on this as well. So it's been really interesting for me, and it helps my mind shift in what I need to do. So I think I need to go and advocate for exactly what we discussed right at the very end. And that comes about by the people asking the right question in the right context. You suddenly see what's needed.
The future for severe asthma is more bright than it ever was.
I'd love to agree. And I'd like to leave the last word with Olivia, because she's what we're all engaged with this for.
Thank you very much. And I think what's highlighted in today's conversation is the necessity to link the basic science with the sort of the more applied science, bringing it all together to really hit that end goal for patients. The other thing is not stopping. Just because something didn't work, it's understanding why it didn't work. For me today, it's been really inspiring, because speaking to someone like yourself, it just gives you hope that, yes, there's people out there doing things, people out there who are dedicating careers to making people's lives with severe asthma better. And all I can say on behalf of everyone is thank you.
Thank you.
If you'd like to hear more podcasts about the work being done by SHARP from the perspectives of different stakeholders, then visit the SHARP website at europeanlung.org/SHARP. You can also find additional information about SHARP project and research programs, as well as how to join the SHARP CRC by visiting the website.